Hi, I am Dr.T.SUMATHI, Department of Zoology. Right now, I am taking the subject Immunology. Already you know very well about what is meant by immunology. We have the antigen and antibody in our body. So, we have to know about the immunology. In this shows, we have to know about the Unit 5. What are the immunotechniques available? One of the techniques also ELISA, and second techniques also Radioimmunoassay. We have to know about these two techniques in this slide. First, we know about what is meant by ELISA, what is the uses of ELISA, why we have to know about this one, what is the applications. ELISA means short abbreviation, enzyme linked immunosorbent assay. Why it is important for enzyme? Our body also has a lot of enzymes. Each and every enzymes also participated for digestion and also absorption, all the activities in our body. Our mouth also have the enzyme that is called tyalin. In taking of the food material, first digested by the tyalin, after that it goes to mouth, esophagus, stomach and rectum. That is, you participated so many enzymes in our body. So, the enzyme also very important. Normally, ELISA is a plate-based technique to detect and quantify the peptide, antibodies, protein and hormones. What is meant by peptide? 2 amino acid is combined with the peptide. Antibodies means normal immune system occurs in our body, that is called antibody. Any one of the antigens is attacking our body, that time also they have to secreting the antibody. Proteins, you know very well about the protein. Our body also made up of protein. All the enzymes also made up of protein. And hormones. Hormones also one of the important secretions in our body. ELISA is the basic assay techniques known as enzyme linked immunosorbent assay, and also it is carried out to detect the measurings all the peptides, protein. It's also which one have it is important for the blood sample. We have to identify through blood samples. Normally antibodies are blood proteins. It is produced to respond to one of the specific antigens, and it helps to examine the presence of antibodies in the body, and it has some certain infectious diseases also identified through antigen. So many infections affected the body, bacteria, virus, another one is here, some protozoans also. Most of the virus and protozoans is a critical condition, very pathetic condition to go to our body and affected the system, inner system of the body. That time also we have to identify what are the pathogens affecting our body. These ELISA tests also distinguish to analyze the antibodies assay, and both qualitatively and quantitatively. It is a separation of non-specific and specific interaction take place with the serial binding in the solid surfaces. Normally, it is a polystyrene multiple plate also used to identify the ELISA test. Principle of ELISA. ELISA works on the principle, the specific antibodies, the bind with the target of the antigen, and detect the presence of quantity of the antigen binding. For example, anyone affected anyone of the dengue or AIDS and that disease, the dengue creating virus where it is present in our body, they have to identify through ELISA test. After that, they have been given the antibodies of any one of the person. They have to binding with the antigen, the killing of the antigen. The ELISA can provide a useful measurement of the antigen antibody concentration. After, what are the types is available to identify the ELISA? There are 4 major types to identifying the ELISA test. First method one also direct method, indirect method, sandwich method, another one is a competitive ELISA method. What is the difference between these methods? First, direct ELISA method also, antigen is coated the plate after screening of the antibodies. After indirect method also, antigen is quoted and also they have to screening both antigen and antibody. In sandwich ELISA method also, antibody is quoted, not antigen, and screening of the antigen also. In competitive ELISA method, screening of the antibody. After that, we have to identify in the ELISA method. This picture shows direct method, indirect method, and sandwich ELISA method, another competitive ELISA method. This method, direct method, Y shapes also you know very well about the antibody. The primary antibody is a conjugate method. After indirect method, and secondary antibody is conjugate that method. Third, sandwich method, capturing the antibodies and also inhibit the antigen in the competitive ELISA method. This method also indicating the antigen-antibody reaction in the blood samples. This is a procedure of ELISA method. Normally, ELISA is one of the easiest blood test that can be carried out in comparison to others. Why it is easiest way? It gives a rapid result and quick and recovers the blood samples of the patient. The entire procedure of the ELISA is mentioned. The antibodies is attached to the polystyrene plate and already solid surface. And also it attracted the affinity towards the bacteria and other antibodies and hormones. A microtiter coated with antigen. Microtiter means that is a plate, ELISA plate, also coated with antigen and also filled with antigen-antibody mixture, after which free antibodies are removed by washing. Free antibodies mean excess of the antibodies also are removed. Another second antibody, the specific of primary antibody is added, which usually conjugated with the enzymes. Next one, a free enzyme is linked, the secondary antibodies are removed by washing the plate. Finally, the substrate is added. Which one we have to identify? That is called substrate. We have to add. The substrate is converted to the enzyme to form a colored product, after which can be measured by using the spectrophotometry. The colored substance is depends upon the different type of bacteria and different type of virus, also identifying the staining method, that is called colored spectrophotometry. What are these procedure also? Normally protein, HCC protein, which indicates the pregnancy to detect the ELISA. This is a combination of the blood and the urine sample also purified. The enzymes normally added in the system. In this accent of this hormone protein in the sample, the only enzyme is linked to bind the solid surface. The more the substance is interest is present, and the more reaction take place to link the enzyme to bind the solid surface also. These reaction normally indicating the usual changes in the color of the solution, because the color of the solution we have to identify which pathogen is affecting the body. Of what are the disease we have to diagnose by using the ELISA. So many disease also we have to diagnose. First one is the ELISA. It can be used to detect the some using conditions. Ebola, one of the virus, that is one of the disease we have to identify by using ELISA. Precocious anemia, anemic, if any person is affecting anemia, we have to identify through ELISA. Another one is the AIDS. You know very well about that person. ELISA is one of the main tests. Everybody is known about the AIDS. That is one of the severe disease. We want to affected the disease. We have to know about the ELISA test, identifying this person is affecting the AIDS, rotavirus, Lyme disease, syphilis, toxoplasmosis, Zika virus, carcinoma of the epithelial cell. Everything also we have to identify through the ELISA test. What are the advantages of the ELISA? The ELISA, what is the uses of this ELISA test? The result is fetched the ELISA to give the accurate diagnosis of the particular disease. Since two antibodies are used, one can be carried out the complex sample, the antigen is not required and purified to detect. Another one is a highly responsive, since direct and indirect analysis method can be carried out. Normal, another one is it is a rapid test method, already I told you. The yielding of the result also very easy. It is quickly we have to intaking of the result. Possible detection of the ELISA and the ranges from quantitative, semi-quantitative, standard curve, and qualitative calibration curve model. Everything also we have to identify. Normal easier to perform and uncomplicated process as compared to other assay which require the presence of radioactive materials also. And the applications of the ELISA, the presence of antibodies and antigen in sample can be determined. It is used to the food industry, detect the food allergens is present, to determine the concentration of the serum antibody in the virus test, and also it is very useful to identify the disease outbreak, to evaluate the spread of the disease. Example, during the COVID time also, we have to uses of so many ELISA test identifying the person is affecting COVID. The rapid test kit being used to determine the presence of antibodies occurs in the blood sample. Once affected this COVID infection, immediately the person is doing the ELISA test. This plate, ELISA microplate, this plate also used to identifying the protein. The samples also coated in the well of the micropipette. After the identifying the techniques of the ELISA, the protein also loaded. This ELISA test, we have to know about the concluded of ELISA. It is a very sensitive and robust technique, the vast application. The techniques used to specify of the antibodies, interaction, and it is a very sensitive enzyme also we have to identifying, and detecting the low as picomolar concentration, and various antigens also, antibodies also identifying depends upon the given sample. This technique is based on the immobilization of antigen and addition of the enzyme is labeled correctly. We have to specify the particular antibody to specific the antigens. It's very important. And addition, the enzyme substrate also binding. You know, already you know really about the lock and key system. The substrate and the enzymes also attracted. After that is a lock and key system. It is based on the enzyme link. The presence of antigen is detected by the colored product of enzyme reaction. There are different type of ELISA with varying sensitivity to application. The ELISA is identifying the diagnosis, the medical field also. The cancer screening also very important, and also drug identifying, drug testing also very important. And many research we have to using ELISA test, and the food industries and many more industries also identifying. In food industries, we have to identifying the allergies of the body, that is a sensitive of the food. Which food is creating allergy, we have to identify through the ELISA test. This is a conclusion. After that, we have to know about another technique that is called radioimmunoassay. This radioimmunoassay is shortly called RIA test. This full form RIA is called radioimmunoassay. The assay uses a highly sensitive method. Comparison ELISA also very sensitive method to determine the antigen is concentration of the sample. This technique first introduced by Rosalind Allo. That is, person is a nuclear physicist, and she developed this radioimmunoassay together the doctor, one of the Dr. Solomon person. After that, they have to developing the RIA used to measure the small concentration of substance in the body, such as hormone in the blood, not enzyme, the hormones in the blood. Normally, yellow and the person, the two person is tracked the insulin in our body by injecting the radioactive iodine into the patient's blood. After they have to identifying this particular hormones is present in the, normally insulin is also one of the hormones. It's very important of the, you know very well, very important of the diabetics also. What are the basic principles of RIA? This method evaluates to assay or material to determine the particle is present, otherwise is unmeasurable or detectable only difficulty. So, we have to identifying the assay of the hormones. When unlabeled antigen from the standard or samples is allowed to react with the set of the some amount of the antigen, that is very tracer amount. Normal decreasing of the quantities of tracer are bound to the antibodies. the amount of unlabeled antigens also rise. Competitive binding in the fundamental concept of radioimmunoassay, in which radioactive antigen is compete with non-radioactive antigen for a given number of antibody or receptor binding site, and also it uses after some analysis. In other words, the binding of the unlabeled antigen to the fixed to definite amount of antibody to introduce the displacement of radio-labeled antigen. That's also very important. Now, which it is reduce the radioactivity of the antigen-antibody complex. So, the RIA help in the determining the concentration of such labeled molecule by measuring the radioactivity rather than the chemical activity. What are the application of radioimmunoassay? This approach, it is a substantial influences of the medical diagnosis because of simplicity, which the test may be performed while ensuring it is very accuracy and specificity, and also it is a sensitivity. Normal radioimmunoassay can be used to detect the substance like hormones, vitamins, serum protein, infective agents, any one of the drugs affected the body, and also it can detect the substance, the range of very small particle, that is a nanogram and picograms also. These small amount of particles also we have to identifying by using radioimmunoassay. Any one of the person is affecting preliminary, we have to identifying this particular RIA test. What are the type of RIA is available in the field? Double antibody RIA. These RIA, another one is a coated RIA. The double RIA, antibody RIA is a coated tube. The two most prevalent RIA techniques used to drug detecting the biological matrices. These tests also identifying the second antibody is added and doubled the antibody of the RIA aid, and also formation of precipitation of the blood. It is bound in the main antibody of the body. The unbound labeled medication can be easily to remove the primary and secondary antibody. The antigen complexes also the time precipitated. So we have to identify these particular antigens is affecting this one. The antibody also secreted immediately. We have to identifying particular antigens also. Another one is a coated tube RIA. The primary antibody is coated on the interior of each tube, and the coated tube RIA by draining out the supernatant also. The unbound labeled medication may be readily removed. After the time, what are the types of RIA? Evaluating the gamma counter to calculate the counts per minute, which are inversely proportional to the amount of drugs contained the original specimen. After the radioimmunoassay are sensitive and specific, but they need to particular handling and disposal of the radioactive method. This radioimmunoassay method is important for identifying by using the radioactive material. What are the advantages of the radioimmunoassay? So many different advantages of the occurs. It is normally very sensitive test, and it can detect the antigen of the picogram levels. The antibody also antigen is response is very specific. Hence, the test is highly specific. Due to this high specificity, we have to measuring the single hormone to present in the presence of other hormone. Any one of the hormones is increasing in the body, any one of the hormones is decreasing, other secretion level decreasing in our body. We have to identify through the RIA technique. The RIA can be used to measure the hormone variety of the body. In case the hormone occurs in the fluid, including in the blood, sometimes it goes to the urine and it goes to the saliva. We have to identify where it is present, the hormone, what are the uses, and also they have to differentiate from that ones. A vast number of samples can be proceeded through this RIA technique, such as any one of the blood samples, urine sample, and the saliva samples also identify these particular things, the particular saliva, particular hormones is occurs in the body, and also it can indirect analysis method also. This method also uses up to identifying the all the hormones in the body. The process is required is highly specific and actively radio labeled the hormone. After that, the radio labeled, we have to identify the particular hormone is affected, these particular ones. And normally it is a scintillation counter is required. This counter also fully radioactive isotope occurs, this counter, scintillation counter, and they might not be easily available. So we have to identify through the RIA techniques. While carrying out of this process, the testers need to be extremely careful as they will be handling the radioactive component. Because sometimes the RIA technique is sophisticated to time-consuming method. It is not appropriate of routine testing. RIA also costly. Normally, the equipment and reagent is necessary to RIA might be the prohibitory pricey. And the formation of equipments and we have to using of the reagent also costly. Furthermore, the RIA is operated by qualified individuals. It is not usually available in the many places, such as small hospital and laboratory also. So, in the highest level of the laboratory and the highest level of the hospital or the hospital also avails this RIA technique. So, we go and testing that particular places also. This is one of the steps we have to following the radioactive antigens is bounding the attraction to the prepared. This is the antigen-antibody reaction. We have to use of the method. The patient is normally antigens, the blood samples, it also added in the second one. Unlabeled antigen from the patient, the blood also added. Step 3 also radioactivity labeled the antibodies, and also we have to identifying and displaced. Step 4 is unbound the antigens and separate the radioactive antigens, and we have to identifying the antibodies and we have to measuring it. This is a basic method. The picture also explaining the ELISA method. The ELISA detects the antibodies, the antigen complexes using the enzymes, whereas the RIA detects the antibody-antigen complex by using radioisotopes. The ELISA involves the labelings and antibodies, whereas the RIA involves labeling antigen. This is a difference between the RIA and the ELISA. ELISA detects the enzymes, where the RIA detects the antigen-antibody complex during radioisotope. Enzymes also very important in the ELISA. RIA is mainly concentrating only the radioisotope, not enzyme. So already I told you the enzyme identification is a very rapid and also very useful in the ELISA test, and we have to obtaining the result also very quickly. But the RIA test detects the antigen-antibody the same but complexes, because we have to use this radioisotope. It is expensive compared than ELISA. Normally ELISA involves the labeling the antibodies, whereas the RIA involving labeling the antigen. And the we have to ELISA labeling the antibodies only. In RIA is involved to labeling the antigens only. After the ELISA, easily to less sensitive and when compared to the RIA assay. ELISA easily less sensitive means we have to easily identifying through the techniques of ELISA, but RIA is a very little bit difficult to assessing the RIA radioimmunoassay techniques in comparison ELISA test. That is here we have to use of immunologically two techniques also very important to identifying the any pathogens affecting the body. It is mainly identifying the antigen-antibody reaction and the antigens how they activated the body. Where it is present in the blood samples, urine sample, we have to identify protein, peptide, anywhere, identifying these two methods, and it is uses for the people for easily cure the disease. That is a expensive, one of the expensive method of RIA, but it is a very useful to identifying easily. We are following this method to identifying the disease, the antigens, and we have to escaping from the disease. We live in the normal life. Thank you.